Hello guys, I am Shubham from ShubhVed Tutorials. In this video, we will solve part C questions. The remaining questions we will be solving. Till now, we have solved 20 questions of the CSIR NET June 2025 exam. So before we start, subscribe to our channel and press the bell icon to get notified whenever new results. This is absolutely free from your side, but it will help us grow. First question, two samples were run on a flow cytometer and the data were plotted as a histograms A and B as shown below. The following statements were made. Figure A represents three normal distribution with more variation within groups than between groups. Very easy, सिर्फ देखना है डायग्राम एंड आंसर करना है. तो फिगर ए में यू कैन सी जो ग्राफ्स है ब्रॉड है, राइट, जो पिक्स दिख रहे हैं इंडिविजुअल पिक्स रिप्रेजेंट इंडिविजुअल ग्रुप. So the groups have more variation than between groups. मतलब between groups में gap नहीं है, there is a overlap, right? जो peaks है overlap हुए हैं. So yes, this is correct. Now about figure B. Figure B represents three normal distribution with more variation between groups than within groups. You can see here यहाँ पे between group gap है बहुत. So yes, variation between group ज़्यादा है than within group. Within groups you can see individual peaks narrow है. So this also option is correct. Okay, so A B हमें चाहिए, option 1 and option 3 eliminate हो जाएगा. Okay, then statement C is ANOVA F test is likely to be statistically significant when applied to figure B but not figure A. This is also correct. ANOVA F test, ये जो between group variation होता है as we can see in the figure B, उसके लिए ज़्यादा significant होता है, right? So this is correct. A, B, C, option 4 correct होगा and not option 2. I will tell you here the formula also. F is equal to ANOVA F जो है between groups upon within groups. Okay. The following statements are made about transgenic mice technologies. Catalytically inactive Cas9 fused to a transcription activator can be used to activate specific gene expression. Yes, correct है. CRISPR Cas9 हमने डिस्कस किया है. I will add that link in the I button. Inactive Cas9 is basically dead Cas9. Dead Cas9 means उसके nucleus domains inactive है. Okay. So if this is fused with transcription activator, this can activate a specific gene, right? Gene activate हो सकता है. Okay, वहाँ पे न्यूक्लियस तो काम नहीं करेगा, मतलब कुछ कटिंग वगैरह नहीं होगी, मॉडिफिकेशन हो सकता है. तो A is correct. Using CRISPR-Cas9, a precise gene replacement in a target locus can be accomplished by non-homologous and joining repair. Wrong है. Precise gene replacement non-homologous and joining से possible नहीं है. The next pathway जो है homology directed repair, उससे precise gene replacement होगा. उसमें हम donor template को inject करते हैं along with the CRISPR machinery, right? So B is wrong. अगर B wrong है 3 and 4 eliminate हो जाएगा. And when B is wrong, D is also wrong, right? Next, using CRISPR-Cas9, many genes can be targeted at once by expressing many guide RNAs in the same cell. Yes, correct है. Okay, मतलब एक ही टाइम हम अलग अलग जीन्स को टारगेट कर सकते हैं by expressing many guide RNAs. So yes, this is possible. Next, all chimeric mice derived by injecting engineered mouse embryonic stem cells into blastocells will carry the trans gene in the germline. Wrong. All extreme statement है. All mice will not carry the trans gene. Right. जैसे कि हमें पता है रिकॉम्बिनेंट्स भी all cell recombinants नहीं होते. Right ना? So this is wrong. Okay. Already D wrong है. In the Cre-Lox conditional system, the Cre recombinase is inserted into the gene to be knocked out. Wrong. Cre recombinase को insert नहीं करते, LOX P sites को insert करते हैं. Okay, around the gene. LOX P sites where Cre recombinase will act. Right? So this is also wrong. So E we don't want. Option 2 answer होगा. Got it? Following are few statements regarding mapping population and principles of genetic mapping in crop species. A. For self-pollinating species, both F2 and F1 derived haploids, double haploids, can serve as mapping population. Correct है. Okay, mapping में इसका role important है. F2 and F1 derived haploids. For cross-pollinating species, F1 population can be used for mapping studies. Yes. Right? जेनेटिक्स में हम देखते हैं F1 क्रॉस F2. That is important. Bulk segregant analysis is often used to identify linked markers for complex traits with multiple genes and environmental influences. No, wrong है. This is used for monogenic. Okay, single gene. Basically qualitative inheritance. Right? So C wrong है. C present everywhere. C. We have to choose the incorrect statement. Right? A भी correct है. Eliminate this. Okay. Next, in crop plants, sets of different breeding lines can be exploited for trait mapping. Correct है. This is correct. Breeding methods such as single seed descent and pedigree selection promote genetic variability. No, genetic variability नहीं होती इन methods से. E is wrong. C and E incorrect statements है. Option 4 answer होगा. Okay. Match the following very easy to solve. Okay, technique से है. Given below are names of techniques in column X and their characteristic features measured for molecular characterization in column Y. X-ray crystallography, diffraction, right? DNA structure determination में इसका बहुत रोल है, right? A3 option 1, option 4, 2 and 3 will get eliminated. Then colorimetry, absorbance, right? Principle है colorimetry का absorbance. E7 option 1, right? ऑप्शन 1 ही आंसर है. NMR स्पेक्ट्रोस्कोपी, केमिकल शिफ्ट, जो केमिस्ट्री है मॉलिक्यूल्स की, दैट वी कैन अंडरस्टैंड बाय NMR. रमन स्पेक्ट्रोस्कोपी, वेव नंबर. ओके, सो B5 T4 and CD spectroscopy electricity, D6. Right? Last question. A polymerase chain reaction PCR with all necessary agents was carried out to amplify a 200 base pair target region. ये बहुत इंपॉर्टेंट है. I will highlight this. On a human chromosome, Y chromosome, PCR was started with 100 copies of double-stranded DNA as a template. Okay, so double-strand DNA को use किया है, 200 copies are used, right? And there is a target region given of 200 base pair again. Sorry, here 100 copies. 100 copies, right? So now this target region we want to amplify. Okay, we want to amplify target region. Assuming 100% PCR efficiency, how many copies of the desired 200 base pair fragment consisting of only the target region would be produced at the end of third PCR cycle? दिया है third PCR cycle. So टारगेट रीजन हमें देखना है सिर्फ ओनली आफ्टर पीसीआर फर्स्ट साइकिल लॉन्ग प्रोडक्ट्स आर फॉर्म. ओके, लॉन्ग प्रोडक्ट्स बनेंगे. जो टारगेट रीजन का है बनेगा ही नहीं. यू कैन सी हियर सिर्फ लॉन्ग प्रोडक्ट्स बनेंगे. आफ्टर सेकेंड साइकिल देयर विल बी लॉन्ग प्रोडक्ट्स as well as single strand products. Okay, single strand DNA हमें मिलेगा of a target region, right? Because ये extend होगा, right? तो single strand हमें मिलेगा. After third cycle, we will get the double strand DNA of this target region. Okay, and हमें third cycle का ही पूछा है. So after third cycle, we will find the double strand DNA of target region, that is 200 base pair DNA. Okay, now if I tell you by using a formula, there will be a formula here, 2n minus 2 bracket n. 2n यहाँ पे 3 है, here also there will be 3. So here it will be 8, here it will be 6 and इसका होगा 2. Okay, 2 है. So 2 DNA molecules are produced after third cycle. Now जो starting material है कितने copies है? 100 copies. Multiply with this 2. Okay, and then you will get 200. So 200 होगा इसका आंसर, right? So ऐसे टाइप के क्वेश्चंस जो है, this is how you can answer, right? Of the target region if they have specified. First double strand product you will find third cycle. First single strand product you will find at the second cycle. Remember this. Okay, तो इस वीडियो में हमने 5 क्वेश्चन सॉल्व किए एंड प्रीवियसली 20 क्वेश्चंस. तो 25 क्वेश्चंस वी हैव सॉल्व एंड पार्ट सी में 25 ही करने होते हैं. I have shown you questions from the content available on our channel and also using some basic knowledge information you can answer like this man the following. Okay, सिर्फ 2 options से आंसर हुआ. So that's it for this video. I hope this video helps you. If you find it helpful, do let us in the comment section. Like this video, share this video with your friends, subscribe to our channel and stay tuned for the upcoming videos. Thank you.